my god, what happened to it?
Well, Jessica, that frog's cells contain a strange number of chromosomes called aneuploidy.
Wait, what?
Do you guys remember about meiosis?
Yeah, that's where a diploid cell divides into four daughter haploid cells.
Well, that process isn't perfect. Occasionally, a mistake occurs when the chromosomes are separating. What can happen is called a non-disjunction event. This is where, in either metaphase 1 or 2, the chromosomes don't separate properly. So, one of the two daughter cells ends up with an extra chromosome, and the other ends up with one less. This actually happens fairly often. In fact, scientists believe that up to 25% of all human egg cells are aneuploid.
I don't understand. Does that mean my cell could be aneuploid?
No. Most of the time, these cells are unable to survive and be fertilized. However, occasionally they do. People with Downs, Klinefelter's, and Turner syndrome are the product of an aneuploid cell.
So wait, relating back to what we talked about at the beginning of the semester, a cell which is aneuploid has decreased fitness?
For the most part, yes. However, there have been instances where this could actually increase a cell's fitness.
Wait. Now you're just confusing. You just said most of the cell doesn't get fertilized.
In a recent paper published in Nature, researchers looked at the effects of aneuploidy on the yeast Saccharomyces cerevisiae. They observed that in some cases, the aneuploid cells were able to survive in some conditions that the normal cells would not.
How do they manage to test that?
Well, first they began by creating aneuploid cells. They started by inducing meiosis in yeast strains with an odd ploidy, like 3N and 5N. They karyotyped the different cells to identify which are aneuploid.
Karyotyped?
Yeah, they looked at the karyotype, the number and appearance of the chromosomes in the nucleus. Honestly, don't you read the textbook?
Hey, I've been busy, God.
Anyway, Jessica's right. They then used these cells in their experiment.
What sorts of experiments?
By exposing the cells to different conditions, they were able to determine how the aneuploid affected the cell's fitness.
Then how did they measure the fitness exactly?
By measuring the average protein levels in the cell.
So they just measured the protein level, and that's it? How do they know if fitness increased necessarily?
Well, they compared it to a normal cell, of course. They selected 38 strains with aneuploid, which were labeled A1 through A38, and placed them on a dish with 3 euploid strains, labeled U1 through U3.
And then they exposed them to various conditions and tested their fitness.
Yes, they changed temperature and pH, and even exposed the yeast cells to antifungal medications.
Oh, I see now. So here's the image for the control experiment under normal conditions.
And look, here's one where they lowered the temperature to 16 degrees Celsius.
Yeah, you can see there are much fewer strains doing well, and you can clearly see that the majority of them, especially the control strain, have a drop in fitness.
And this last set of data was from exposure to bleomycin.
Wow, look at that. Those few aneuploid cells did really well compared to everything else.
Yes, they did. But remember, overall aneuploid cells have decreased fitness. This graph visualizes it well. On the left graph, you can see the various strains of aneuploids used. The red indicates that that particular chromosome in the strain had more copies than the euploid. The blue means it had less. On the right, this showed how the fitness compared to the euploid. Red means it did better than the euploid, and blue means it did worse. The darker the color means the greater the difference in the two fitnesses.
Oh yeah, so I can see for the most part it's just blue, but look there. There are a few with really, really dark red indicated.
What are some possible reasons for the increased fitness?
Well, Jiyoung, especially for the increased fitness to antifungal medications, they speculated that overexpression of certain genes created a hyperresistance.
Wow, that's so cool.
It's like seeing evolution before our eyes.
Indeed. Now, let's continue on with the lesson. Alright, well, continue this on Wednesday.